Case Western Reserve University's Great Thinkers series proudly presents the Origins Science Scholars Program. These lectures are presented by the Institute for the Science of Origins, a partnership of Case Western Reserve University and the Cleveland Museum of Natural History, with the generous support of Richard Morrison and the assistance of Case Western Reserve University's College of Arts and Sciences, Media Vision, and WVIZ PBS and 90.3 WCPN IdeaStream. Welcome to the Origins Science Scholars Program, a series of talks on current research topics in origin sciences. Hello, my name is Glenn Starkman, Director of the Institute for the Science of Origins, and I'm pleased to serve as your host for the series. The talk you're about to watch is entitled The Future of Human Health. In this talk, Eric Artz, Professor of Medicine at Case Western Reserve University and a Fellow of the Institute for the Science of Origins, will tell you about global infectious diseases as the greatest health concern facing humanity, with an emphasis on HIV/AIDS. Please enjoy the talk.
I'm very happy to see so many of you out here tonight. It is nice that the sun is shining, so to be actually locked away in a room without sunshine right now is something to miss, the outdoor views. So I was given a topic tonight that I felt a little intimidated by. Glenn Starkman asked me to present on the future of human health, which in some aspects is a very large topic, and I have taken the liberty of sort of bringing that down a step. And the way I'm going to focus this talk today is talking about global infectious diseases with an emphasis on HIV. And I start this by saying that it really is our greatest health concern. Many of you know a lot of the stuff I'll present today, but I hope that I can introduce you onto some other aspects. So, infectious diseases have always been a part of our history. It's one of the ways that we actually evolve in many different instances to new infectious pathogens. It allows us a level of protection from previous pathogens, like, for example, influenza. But every once in a while, something comes along that creates a horrific epidemic in the human population. And all of us are quite familiar with the plague in the 14th century, which is always shocking to me when I put presentations like this together at the level of devastation that occurred. When we think about close to 20 to 45% of the human population being wiped out by pestis is quite surprising. But we've had many other outbreaks of large epidemics of a large scale. For example, in Cairo, again, 13% of the population succumbs to cholera. There's many cases of dysentery, for example, during the Crimean War, and in the Boer War. 5 times more people die often of these infectious pathogens created by the war conditions than the actual war itself. So throughout the talk this evening, what I'm going to do is give you a series of introductions of pictures. And what I want you to do as I'm going through this is just yell out in the audience when you know what I'm referring to, okay? So it's a contest, all right? So you win something, I don't know what yet. All right, so this is a very old graph, probably done on original graph paper, definitely not done with Microsoft Excel. So this is looking at the influenza pandemic in the 1918s and 1919 Spanish flu epidemic, when estimates range from anywhere between 10 to 20 million people to as many as 30 million people dying of that flu. And here you can see how the flu just took off in different parts or different regions of the world, reaching caseloads of close to 30 to 40 people infected per thousand. This is just the UK mortality over this period of 1918 to 1919, and you can see this incredible rise in the flu epidemic and then another peak occurring here. And of course, this is what this devastation causes. Incredible effects on the healthcare system, the management of these patients, and then healthcare workers themselves getting sick. And this is one of my favorite observations of closing of movie theaters, closing of public meeting and meeting places and gathering places just to avoid any contact with individuals. And this is in a situation when we know that these are infectious pathogens that can be spread through human contact. That's not the case when we were looking at the bubonic plague, for example. So why am I discussing infectious diseases in the future of medicine? And one of the reasons is that really infectious diseases is the largest global killer around the world today, and probably has been through the ages. We see a rise in cardiovascular disease, cancers, and many others, but infectious diseases accounts for the vast majority of deaths in the world today. And when you account for the fact that a lot of cancers are caused by pathogens, and even cardiovascular disease, it really is the majority. This becomes even more stark when we look at children between the ages of 0 and 4, where pathogens are the major killer among our youth. So we have some major pathogens that are affecting this mortality rate. This is a combination of probably close to 20, 30, 40 pathogens that make up this huge amount of respiratory diseases that cause a large amount of death. But a good percentage of this is still caused by influenza in the world today. But our three biggest killers, again, are AIDS, TB, and malaria. Diarrheal diseases, again, are caused by many different pathogens. But three distinct pathogens cause at least half of the infectious disease deaths in the world today, and probably at least a quarter of the deaths worldwide are caused by any disorder. So here's the first exercise for you as we go through this talk. I'm going to show you global distribution maps of these diseases, and I want you to figure out which disease applies to which global distribution map. So here's the first one. So just make a mental note. You don't have to shout these out. Here's the next one. And here's the last one I'll present. Okay, do you want me to go back through them? There we go. 1, 2, 3. All right, so if you guess the red here is malaria prevalence, you're right. And not surprising, this is really through the tropical zone. You can really just map out the equator, the Tropic of Capricorn, and the Tropic of Cancer, and that's really where malaria concentrates. Although malaria had been known to penetrate different regions of the temperate world, unfortunately, or fortunately, a lot of prevention programs have prevented these seasonal outbreaks of malaria. Now, tuberculosis is occurring in more of the tropical climates, but again, you can see it increasing all over the world, specifically in Russia, where up until recently they've had more major health concerns based on getting care out to patients. And then HIV prevalence, again, is spread across the globe, but with its largest concentration in the subtropical and tropical regions. And the only exception of that is what's happened in South Africa, which I will discuss a little later today. Now, I find this always striking because you can map these infectious pathogens based on the GNP worldwide. Basically, the colors are now opposite. The people that earn the least amount of income is where these pathogens really persist. And surprisingly, when we look at malaria and tuberculosis, these are two diseases that we can basically cure if we had enough money and the greatest amount of ability to penetrate these low-income countries. And even in regards to HIV prevalence and HIV disease that I'll discuss later, this is where we could also provide effective health care, prevent disease progression, and prevent new infections. But again, this takes a global effort and considerable funds to initiate this. Okay, this is not as clear, but just from the simple impact of these different pathogens and pandemics, we have an issue that basically it's costing the globe already considerable amounts of money. And we don't have the SARS figures up here, but SARS was such an economic condition for Southeast Asia and China that it's created a whole infrastructure of the Center for Disease Controls in China to deal with these epidemics. So this is a quote that I quite like, and I'm just going to read it to you because I can't really paraphrase it. So microbes and vectors swim in the evolutionary stream, and they swim faster than we do. Bacteria reproduce every 30 minutes. Viruses sometimes even faster. For them, a millennium is compressed into a fortnight. They are at fleet of foot, and the pace of our research must keep up with them, or they will overtake us. Microbes were here on Earth 2 billion years before humans arrived, learning every trick for survival, and it is likely that they will be here 2 billion years after we depart. And I can't stress this more, and I'm sorry if I'm getting on a soapbox now, but really this country and the developed world is progressing because we invest and we work hard in research and development. And this research and development help to find cures, to find preventative strategies, and really, hopefully, eradicate these infectious pathogens. But we're really at a breaking point right now because funding is being cut quite dramatically in these areas. Okay, so what are our current microbial threats? Well, there are newly recognized agents like SARS, Acinetobacter. There are mutations of original zoonotic agents that cause human disease. We know the classic examples of flu. And there are others as well that are coming on board, and we always have to be fearful of. Okay, so in the last couple of slides, I want you this time to yell out when you recognize the pathogen I'm going to show you. So these are new and emerging pathogens, and can you name them? Anybody? I'd be really impressed. I'd actually have to leave if you guys could name it at this stage. Okay. Who said SARS? Come on, yell it out. I can't identify you. Oh, over there. Okay, excellent. Yes, absolutely. And this is my favorite picture. Yes, in 2003, we had a SARS epidemic. And I'll show a little figure later about showing how travel plays a role in the spread of these pathogens. But SARS was basically introduced by, oh, and I can't remember the name of the cat. Who remembers the name of the cat that introduced it? Oh. You are no help as an audience, I tell you. Anyway, we'll come up with it at some point. But in just a short period of time, in a matter of months, we had over 3,000 cases in China. We had an introduction into Toronto, a major epidemic in Toronto. And basically, it took all China could do was to contain this epidemic. They shut down apartment buildings, did everything they could do to prevent its spread. And in a communist country, they can do a lot in terms of impeding the freedom of their citizens. Okay, next one. Anybody?
Ebola.
Thank you. So I'll show you some pictures here. So that's Ebola. It's very characteristic. It's an RNA virus. It means it has RNA as its genome. It's one of the longest RNA genomes that exists within a virus. That's me in Uganda sitting in the Ebola caves. I'm still here. That was done 2 years ago. This is a cemetery, unfortunately, in Zaire of patients that were buried with Ebola. And here's someone suffering with Ebola in a recent outbreak. So the important thing to note with this is there's a very low burden of Ebola at any time in human history. So from 76 to 2003, we really are just talking about 1,000 cases. Okay, last one here. Yeah, HIV. So everybody knows the quilt. This is the actual virus. This is a school that I work with in Uganda. This is an orphanage school, and almost all of these children are orphaned because of AIDS, and of course Rock Hudson. And this is just the accumulative climb. So I showed you about 1,000 cases over the last, what was it, about 15 years of Ebola. And this is what our current burden is. In 2007, close to about 33 million infections worldwide. These are 33 million people who will die. Okay, and the pathogens keep on coming. These are new pathogens that have been identified over the last few decades. This one is one of my favorite, the hantavirus, cynombravirus, also known as the virus with no name, that's caused by the deer mouse. It's one of the most pathogenic viruses known to man. It has a mortality rate of close to 50 to 80% once you get infected. So can you name the eradicated pathogen? This is called making Fred happy. Anybody? The eradicated, close to eradicated pathogen? Thank you. Yeah. So, and this elimination is not really due to vaccine, but really due to better hygiene practices and education of the public as to how to prevent leprosy. Excellent. This is related to the vaccine, and this would make Dr. Robbins very happy. But unfortunately, we have a retention of infections in different parts of the globe because of poor penetration for vaccines. And I just mentioned two others where we can say good riddance to. The guinea worm, which is actually a disgusting little insect, parasite. I won't show you any disease pictures because it's absolutely horrific. They kind of roll the worm up onto a stick. And then filariasis as well. Okay, and then the ugly human troots. And I'll just let you read that headline, and I'll take questions related to that. This is the last point I want to make. In Europe already, there's a large refusal of vaccinations of children. And just last year, this is from 2011, there was 5,000 reported cases of measles, and that was the same amount they had for the entire year before.
In one of the first slides you showed, it talks about microbes and vectors. What are vectors as you're using them in that slide? It was at that quote.
Vectors are basically other animal species that carry viruses and then transmit them to us. And then a cycle goes back that we transmit them back to the other animals. And a good example of that is, for example, dengue virus or malaria, which both have a vector that involves the mosquito and then spreads it back to humans and then back to the mosquito, et cetera.
Thank you for joining us. You have been watching Professor Eric Artz of Case Western Reserve University discussing the future of human health. For more information on the Origin Science Scholars Program, please visit the Institute's website at www.case.edu/origins. In the first part of his talk, Professor Artz discussed current and historic infectious diseases, including some that we have conquered or tamed. He also introduced us to some of the disease agents threatening to take their place. In the second part, Dr. Artz will discuss how diseases emerge in the human population. Now we return to the lecture.
Okay, so there are a lot of diseases reemerging, and there are new diseases emerging. And there are many causes for this effect. There are genetic and biological factors. There's new agents that jump from different species into the human population. There's increased survival of humans in one instance that makes us more at risk for other pathogens later in life, for example, when our host immune response is more decayed. There's human behavior, and this is probably one of the major effects. There's political, social, and economic change and sort of incorrect behavior that leads to increases in war, in poverty, and many other conditions that really exasperate infectious diseases. There's physical and environmental factors like climatic change, which I'll give an illustration of how that actually impacts the spread of diseases. Okay, so human demographic change is one that is really causing an increase in infectious pathogen rate. So the best example I can give is that when the Congo Basin, which is one of the origins of many of our infectious pathogens, because that's where most of our primates live, especially the ones most closely related to us, those are the ones that carry pathogens that are most likely to jump into the human species. Back, before the turn of the last century, the contact between humans and these primates were fairly limited. And it was only with deforestation, increases in roads, and further human contact and exploitation that we really saw a major jump in a lot of infectious pathogens in the Congo Basin that then gets spread out throughout the world. It may have been inevitable, but in some ways we can look back and say, maybe we should have done this a slightly different way. I hope a majorly different way, given the exploits of like King Leopold II and such. So here's a population change of Eastern Africa. Uganda is shown prominently above Lake Victoria under UG, and that's where I spend a lot of time. And you can see that the population and density is increasing considerably from the 1960s and 70s to the 70s and 80s. But you also see decreases in the population, which is kind of striking. And I can tell you that some of these decreases in the population in this region was caused by AIDS and complications related to AIDS. But one of the reasons why spread was so much more demonstrable in the more recent times is because population density starts increasing, and then human-to-human contact is much greater. And this is, again, I unfortunately don't have a map of the previous road networks in this part of Africa. But I'm going to contrast this a little bit to what we see in North America. But you can see these are the road networks in different parts of Africa. But you can see that it's really still quite distinct and separate. But you can also see where the contact points of these networks are interacting. For example, in the eastern part of Congo, starting to touch Uganda. And as I'll explain, this corridor is really one of the ones that was majorly responsible for the transmission across Africa and then into the trucking route in various populations of high susceptibility. But really, it wasn't until these road networks were established that we could see transmission in the population. And then I like to contrast that to this is a road network map in the US. So all the black is roads, and you can see it's just about all black. So, and it's important to note that the size of the US is about the size of the map I just showed of sort of sub-Saharan Africa. So that's a very large area, and when you think about the extension of the roads, it's not that great. There's also aspects related to environmental changes and global warming and human behavior that has affected transmission efficiency and has affected the spread of these pathogens. So we have increases in sexual behavior, drug abuse because of increases in population density. There's patterns of outdoor recreation that can lead to transmission. And most importantly, there's social inequality, and there's also inequality of women. So I love bringing this guy up. If you want to blame a tyrant, blame this guy. So this is King Leopold II. So the Congo was really his domain. It really wasn't even a colony. The guy just owned it, he said. And as a consequence, some of the riches of Belgium and a lot of Europe came through this guy. And a lot of it was based on rubber. And excursions into the interior of Congo, interestingly enough, really coincided with the introduction of HIV into the human population at the turn of the century. And I like this description. During the period of 1885 to 1908, millions of Congolese died of a consequence of exploitation and disease. I think more died of infectious diseases than anything. In some of these areas, the population declined dramatically from sleeping sickness, smallpox, and nearly half of the population was devastated, basically because of this guy. Now, I also mentioned that there needs to be an increase in international travel to really spread these diseases globally, to get them into other parts of the world and have founder events that then establish a worldwide epidemic. There's changes in the way we handle food and the way we prepare food, and as I'll explain, that's probably one of the reasons why HIV jumped into the epidemic. So I love this slide. This is from the SARS epidemic, and this is really describing how SARS came to Toronto. So we have two patients arriving on a plane. They go into a household. People visit that household. They transmit the virus. Several of these patients die at some point We still don't know what's going on with this epidemic at this stage. They get into the hospital, and they're put on a ward, and the ward gets locked down. But because other people, they fear, are going to be sick in those wards, they start transferring those patients off the ward to other hospitals, and they isolate the hospital, saying this is the hospital where we're going to deal with this epidemic. But the patients they transferred off of the ward into other hospitals then establish a new epidemic, basically, in those hospitals. And it was a public health nightmare, quite honestly. I had friends who were physicians, ID physicians in Toronto. Many of them got SARS. It was just incredible. And it's really a good example of how not to deal with an emerging pandemic or epidemic. And then, of course, we have the hot zone, right, where we can see classic examples of transmission on airplanes. So what are the contributions, again, leading to this? We have that evolution of pathogens and infectious agents can change, and so they enter the host. They produce new toxins or variants of toxins that aren't recognized by the host immunity, and they cause more devastating diseases, or they have increased transmissibility. We have development of resistance that really allows for the spread, even in the presence of drug, which allows for the spread of mycobacterium or gonorrhea, and as well many other diseases, as we know, related to antibiotic resistance. And this is just an example, again, of where this resistance patterns are emerging. And interestingly enough, here's an example where if the pathogen is found most commonly in the tropical regions, then treatment is also initiated in the tropical regions. We see resistance, but it's not as dramatic as if the pathogen was present in other parts of the globe where there's more access to treatment, where resistance is more prominent. So again, I'll reemphasize, but I don't think I need to spend a lot of time on this, but war and civil unrest is one of the major factors of things like cholera outbreaks and spread of various infectious pathogens because of poor sanitation or breakdowns in the infrastructure. And we see it every time. A natural disaster, war or famine, the first thing we see is an increase in cholera and various other dysentery infections. An aspect of this, I should mention, pooling of plasma. That's another classic example where this has caused major epidemics and outbreaks of HIV and hepatitis because we weren't screening blood products soon enough after we knew the pathogen was the etiological agent, the cause of those diseases. And as a consequence, too many people got infected that shouldn't have. There's a great classic example in Hunan province in China where this sort of illegal blood bank was set up where they were pooling blood and plasma, and suddenly there was this rapid outbreak of HIV in Hunan province that spread dramatically. And then the government, of course, shut it down, probably very forcefully, but effectively. So how do we reduce these threats? We can improve global response. We have the WHO, we have the CDC, and various global response teams, but really they are usually called into action when we have an Ebola outbreak. And they're not called into action when we have an HIV epidemic causing 33 million infections. And unfortunately, this is when we need the best in our global health initiatives to take place and to improve care. We can improve global surveillance. So many people, it's unknown if they're infected or not. A lot of what we see on these maps of prevalence are just estimates because very few people have the facilities, the ability, or the money, or the transportation to get tested. The other thing we can do to reduce these threats is the use of vaccines. For many, many pathogens out there, like polio is a good example, the pox virus, which we've managed to eliminate, but many other pathogens like yellow fever. I'm just skipping on some names, but if you want to really ask where the vaccines come from, Neil is a good person to ask. But they're good delivery systems, but again, we need the healthcare support to get them out there. And then finally, new drug development. And this should not be de-emphasized, because we always paint the pharmaceutical industry as the bad guy in this regard. But without pharmaceutical money, these drugs would never be developed and never be tested in humans and never brought forth. And eventually, they do get to the people that need them the most. Unfortunately, it's often too late. And this is just a good example of the development of antivirals. And I was involved in this, in some of the discoveries, and also putting this horrific chart together with the vice president for infectious disease research at Merck. But this is just basically the development of only 8 anti-HIV drugs that were discovered starting in 1986 with AZT, or AZT, yes, I'm Canadian. And then right now we have close to about 30 drugs in our arsenal that are FDA approved that we can treat patients with. And as I'll indicate later, we need these drugs because the virus develops resistance so rapidly. But you can see that some of these other virus pathogens are also quite devastating and cause global epidemics or affect global populations. But we have been less efficient in developing drugs against them. And you can see the disparity in developing HIV drugs versus other viruses. Again, we discussed already to improve vector control and zoonotic control to prevent this jump from other animals into humans. We need to decrease inappropriate drug use. This goes beyond what is described in this slide, but intravenous drug use is still a major cause of many disease spread. For example, hepatitis is one good example. And then better education is often the most effective preventative strategy. So providing bed nets and telling people how to use bed nets and trying to avoid them from using them as veils for their wedding dresses, which is one of the major black markets of bed nets, as it turns out. Trying to increase education, trying to increase sanitation, get water from a well, not standing water that sits out where you can get increases in crustaceans and guinea worm and many other things. So these are the kind of education programs that are necessary, and they are penetrating these parts of the world. There's many other ways to reduce these threats, but the most important and the most effective is to reduce poverty and inequality.
Can you comment on why Russia looked so predominant in many of your slides, especially Eastern Russia, where the population density I thought was low and they had high population control?
One thing I should mention is when they color Russia in these type of prevalence maps, they color it basically one color, regardless if it's whether in East or West Russia. Although I would have to say that in the eastern parts of Russia, which is still part of the heroin route, some of these pathogens get transmitted very readily.
We hope you've been enjoying the Origins Science Scholars Program with Professor Eric Artz. Professor Artz is director of the CWRU Center for AIDS Research International Corps. We've heard about how human behavior, including globalization, is accelerating the natural processes that lead to the emergence of infectious diseases. He also suggested strategies that we can adopt to limit this process. In our final segment, Dr. Artz will focus on his particular expertise, the story of HIV/AIDS. Now, back to Dr. Artz.
Okay, finally I'm back in my comfort zone, and I can talk about HIV/AIDS. Not that this is a comfortable subject to discuss, but at least I know a little bit more about this than what I've been talking about. And I'll try to entertain you with some pictures since it's the late hours of the lecture, or late minutes of the lecture. So if you get bored, just look at the pictures. They're of my trips to Uganda. So HIV/AIDS. The first question I want to ask is, where did it come from? And where will it go? So it's important to dispel the myths of the origin of HIV, and it's incredible how these myths perpetuate and don't get dispelled frequently enough. First of all, we did not get HIV from having sex with monkeys, and I'm sorry to be so direct on that. these monkeys, these chimpanzees, they live in the high canopy. I've seen them. They're impossible to catch. And they don't fancy us at all, I have to say. So it wasn't some form of sexual contact. And the most likely explanation for HIV transmission into the human population and the form of HIV that's in the simian species is called simian immunodeficiency virus. And that transmission event likely occurred through bushmeat. So in these parts of the jungle in Uganda, in Tanzania, in the Congo, it's really sometimes the only form of meat. So primates are very abundant in those areas, but other animals are not necessarily. So a lot of times they're hunting for these primates. So it often looks very gruesome because we, have a depiction of primates being close to humans, but really it's an acceptable form of meat there. And through the butchering practices that occurs often on the jungle floor, you can see, they got these big machetes, they're cutting up the animals, and it's quite likely that they'll cut themselves some point, and then you could have a potential transmission. And it's important to say that this has probably been going on for millions of years, and the likelihood that HIV only jumped at the turn of the century or the last century is unfounded. It doesn't make any sense. It's probably been jumping into the human population for quite a long time. But again, the population density and the ability of these populations to migrate, or the inability of these populations to migrate, really prevented these from forming an epidemic. So that's just an example of some of the bushmeat trade. So what is a virus? I went through all of this and I really didn't describe a virus. So a virus is a submicroscopic particle, very, very small. A virus does not survive on its own. So a virus needs a cell to infect in order to live and to replicate. So it's an obligate parasite. A virus is surrounded by a core of proteins, and it usually has its genetic material within that core. Sometimes it has a lipid bilayer membrane around it, but basically that's what a virus is. So a retrovirus. What makes a retrovirus unique? I'm asking you. Someone say, I'm going to go blank to blank. What are the blanks? I heard RNA to DNA. Raise your hand. Someone in the audience, excellent, congratulations. You win a prize. Exactly. So RNA viruses are quite prevalent in the world. Influenza, paramyxoviruses, which is another type of influenza, rhabdoviruses like measles, many, many different forms of RNA viruses. Interesting thing about RNA viruses, Bunya viruses, influenza virus, HIV, really they have the same path of initial infection and disease. They generally always cause an acute infection, which results in a huge burst of viremia. It's incredible how many virus particles you will accumulate in the first month or the first weeks of an infection. Sometimes 10 to the 10 virus particles will circulate per ml of your blood, which is just incredible. So when you think about transmission, it doesn't take much to transmit these viruses, and that's why one of the reasons why influenza is so easily transmitted. But the only reason why we survive certain diseases and die of certain other RNA pathogens, viruses rather, is because our immune system comes on in around 2 weeks. And it drives down that amount of virus, sometimes to eradication. And that's how we often survive influenza infections. And it's also the reason why we don't survive infections. So the mortality rate of Ebola is something like 30 to 40%. 30 to 40% of the patients who die of Ebola, their immune systems weren't able to contain the viremia, and they died of hemorrhagic fever. 60% of the patients that get infected, their immune system was great, eradicated it, and really there's little complications associated with the disease after that. And there's very little that separates these diseases. The only difference between HIV and hepatitis C virus is that the immune system controls that viremia, drives it down, but then the virus mutates and survives in the face of that immune response and can survive basically forever because it's got a foothold and it can mutate and stay in that patient until it causes death, cirrhosis, hepatocellular carcinoma, depending on the virus. Oh, so retroviruses are divided into many different families. I'm going to focus on the lentiviruses, because those are the viruses that are most frequently associated with human disease. And lentiviruses are actually found in many, many mammalian species. We have cases in sheep, the Visna virus, the caprine infectious anemia virus, the equine infectious anemia virus in horse. There's a bovine immunodeficiency virus, the feline infectious immunity virus. I'm not a great fan of cats. I'm a dog person. So, but you did see, I don't know if anyone saw that wonderful article that was in Nature Medicine. It was all over the news about these green glowing cats. Anybody see that? Those pretty little kittens with the glow in the dark. Those cats were bred actually specifically to be resistant to FIV, a very unique approach to do it. And I commented to the author, I said, you could make a fortune selling those little fluorescent kitty cats. And then there are almost all old world monkeys are infected by some form of SIV. So the important thing to note about HIV is it's not just one virus infecting 33 million people. It is 33 million distinct viruses infecting 33 million people. And that's one of the reasons why vaccines are so difficult to develop. So if we look at the two types of HIV that circulate in the world, HIV-1 is responsible for 33 million infections, HIV-2 only for about 1 to 2 million infections. HIV-2 would be a serious pathogen if it wasn't compared to its brother HIV-1. But HIV-2 is decreasing in the human population, partly because HIV-1 is overtaking it in certain areas. But HIV-1 and HIV-2 are not the same virus. They share less than 50% sequence identity. That means that the genome of HIV is 10,000 base pairs long, 10,000 ACTGs, okay, in random order. They have the same gene structure, so the genes are in a similar place. And yet, if you look at the sequences of HIV-1 strains and the sequences of HIV-2, they share less than 50% in similarity. I mean, we're talking much more differences than us and ants. So that's the distinction we're trying to make here. So HIV-1 is further subdivided into different groups. There's group M, the main group, group O, the outlier group, group N, the new group, and group P, which really doesn't stand for anything, but they started, using other letters in the alphabet that came in order. M, 33 million, group N, about 5 cases, group O, about 20,000, and group P, only 1 so far. Those different groups, again, share less than 65% sequence identity, so still extreme, probably a difference more than humans and ants. And then even within group M, which is the major cause of the HIV epidemic today, there's as much as 25% differences between each of the strains. So that's dramatic when you're trying to develop a vaccine. If you think about differences in influenza on a yearly basis, if you look at the pattern of evolution of influenza, it is the same phylogenetic pattern, the distinction of the strains, than it is in one patient infected with HIV. So you can imagine that we have to come up with a different vaccine every year to fight off the new pandemic of influenza. Could you imagine that that level of evolution and diversity occurs within a single patient during its disease course? So where did HIV come from? Well, we have a lot of simian species that have natural infections with SIV. A lot of them were acquired through the zoos, so zoonotic jumps. And there's a lot that are not known to have infections. So we can look at pre-existing sequences that we had and try to identify it. I'm assuming that most of you know what these phylogenetic trees are, right? As you move on the horizontal, that tells you the similarity between strains. So if you're looking at HIV-1, and this is group M, for example, this is the distance between this virus, if you move on the horizontal, tracing it back to, for example, this virus. And the length of this line is how many substitutions, how many differences there are in the genetic sequence. So basically, the farther they are apart when you trace them back on the horizontal lines is the genetic diversity between them. So if we look at different populations of chimpanzee species, because we had some inkling that HIV was most closely related to these SIV CPZs, which are chimpanzees, we started looking at the chimpanzee subspecies across Africa. So this is the range of Pan-Trochlidites trochlidites, and this is the range of Pan-Trochlidites swinefurthii, and then there's another subspecies shown here. The swinefurthii, as it turns out, do not have SIVs that are closely related to any of the HIV strains. So we knew, not we, but as a field, knew that we could focus in on that region there. So a very interesting group led by Cameroonian scientists and by an institute in Montpellier, France, basically started looking at different primate colonies, chimpanzees colonies that existed in the Congo Basin in Cameroon. And basically what they did is they sent out their graduate students with big umbrellas to collect poop and pee, so feces and urine. And then they did genetic analyses of the feces and urine to see if there was HIV in the feces or urine. And basically what they could do is identify the virus, and they could then match that virus to the closest HIV relative of that simian virus to the human virus. And by doing so, they could map the potential origins of the epidemic. So what you can find is the sampling that took place in this MB region right here was very, very closely related to HIV-1, whereas sampling in other regions, shown here, just downstream, and basically on the other side of this particular Bumba River, was a different SIV strain that was not closely related to HIV. So chimpanzees don't usually cross water boundaries. So it's quite likely that the chimpanzees shown here were one of the origins of HIV infection into the original zoonotic jumps into the human population, whereas chimpanzees just on the other side of the river may not have had the same human contact. So when did it get here? Like I said, you can kind of trace back the horizontal origins of these trees to a node in the tree. Basically, we know how fast the virus mutates when it replicates. And so by knowing that, we can back calculate the length of these arms on the tree, and branches rather, and then calculate how long it would take to get back to this introduction point. And by there, we can calculate that the likely introduction of HIV occurred somewhere between, now it's estimated between 1910 and 1940. So it's quite a long time ago that HIV has been percolating in the Congo Basin. And what has been done through cumulative sort of dating studies is that we know that chimpanzees were the origin of HIV and was responsible for this huge epidemic that we see here. But we also know that there's another type of SIV strain that somehow jumped into gorillas, and the gorillas were the source of this other HIV strain, group O, that was only responsible for about 20,000 infections worldwide. And then we know HIV-2 came from the Sudi mangabe and was introduced probably sometime around the 1940s. So we have several zoonotic introductions that established epidemics. But where has it gone from there? And I'm going to end with this. I have some other slides, but I don't think I'll have time to present. So we know that all the HIV started in this sub-Saharan Africa in the Congo Basin. And we might have even identified the exact site of where HIV jumped into humans. And this is just showing it here. Now, HIV evolved into these different subtypes, and we'll just describe them as subtype A through J, different letters of the alphabet. Different subtypes have different prevalences around the world now, but they all originated in the Congo Basin, and it was a result of rapid evolution in this Congo Basin that resulted in these different subtypes of virus. And we can date certain spreads of the virus through sampling that was done on patients that unfortunately died of unknown circumstances. And when they have symptoms of immunodeficiency, we can go back and look if they had stored samples. And one of the classic examples is this Norwegian man that traveled from West Africa, again through merchant shipping, and introduced the virus into the familial unit, unfortunately infected his wife, and the wife infected the newborn, and the family died. The interesting aspect of that is that founder event never established an epidemic. It stayed within the familial unit. Then we have examples of early infections, probably in Haiti back in the 1960s. Haiti was involved in several wars and sort of travel and trade with Africa at the time. And then at some point, HIV was introduced into North America, in the US in particular, and probably Canada as well in the 70s. And then from there, it just spread globally. HIV subtype B in particular took root in the developed world, mostly through men who have sex with men, and established rapid epidemics throughout the world. But this continued. It hasn't stayed static, and that's what people don't recognize. All the time, there are new founder events in different parts of the world that establish an epidemic where we had a low prevalence before, and suddenly the HIV takes a foothold, and we start seeing increases in seroprevalence in the population. And a good example of that is in China and India, with the subtype C being introduced probably in the early 90s, and now responsible for millions of infections in that part of the world. And this is a global map, and I'll leave you with that. It's important to state that with HIV, even the prevalence of these different subtypes do not stay static. And different subtypes predominate. I show a lot of yellow on this map. A lot of yellow is this subtype C, which has dominated the worldwide epidemic. If we looked at this distribution of HIV just about 10, 15 years ago, C was almost non-existent. It was a small piece of pie. And now C has come to dominate. And I'll just summarize some of the data that I was going to present on our lab, but I can probably just summarize years of research in just a couple of seconds. But basically what we've done over the last 10 years is we've studied HIV-infected women in Uganda and in Zimbabwe. We recruited these women as participants in our study as part of actually a hormonal contraception study. And they were newly infected. And we studied the natural history of their HIV disease. When they got to the point where they needed treatment, we provided treatment. But we could at least look at how long it took for them to progress to disease prior to needing treatment. And by doing this study, we found some very startling observations. And that is that not all these viruses were created equally. As it turns out, in Zimbabwe, which is shown right there, you can see it's all yellow. That country is predominated by subtype C. And in Uganda, shown up here, which is shown more in this pie chart, subtype A and D predominate. So we were able to compare the rate of disease progression in subtype A, C, and D infected women, probably about 350 of these women, over a 10-year period. And what we found was that not all of these viruses cause the same disease, even though they're all HIV. We found in the subtype C infected women, even though C is spreading globally faster than any other virus, these women were surviving much longer. Their course of disease was about twice as slow as any other subtype infection. As a matter of fact, subtype A and D infected women in Uganda were progressing twice as fast. So the median survival of a woman in Uganda was approximately 8 years if they did not receive treatment. In Zimbabwe, it was approximately 20 years. So I'll leave this as a point for you We also were doing a lot of virology studies, and one of the things we characterized by looking at these viruses and how fast they could replicate in culture is we identified the fact that the subtype C viruses were very slow to replicate. When we put them in competition with other viruses, the other viruses always won, the other subtypes. C was always the loser. So classic survival of the fittest. But if we looked at the ability of these viruses to transmit, all of them were quite similar. So regardless if you're a subtype C or a subtype A or a D virus, transmission efficiency was quite similar. But subtype C viruses were causing slower disease progression, meaning these patients were living longer, and often asymptomatically. They didn't even know they were infected. So I'm going to skip to this. Oh, I should show you this slide just because it's so cool. That's a taxi park in Uganda. I love this slide. So I'll leave you with this question, and I can entertain more questions. Based on what I just told you, if you were even listening at this late stage in the lecture, and I commend you if you were, can you come up with a reason why subtype C is expanded globally in the epidemic, given the fact that it's causing slow disease progression, but also considering that it still retains effective transmission efficiency going from human to human to human? And I'll just end by saying bye and thanking a number of different people. Especially want to thank Roger Bettles, who really provided a lot of slides for the initial part of this talk. He's in the Institute of Public Health at UCLA. He puts together wonderful lectures. This is a team of scientists that work with me on various HIV studies around the world. Our research is largely focused on HIV evolution and looking at drug resistance and how these viruses are causing disease. So, thank you.
This lecture is part of the Origins Science Scholars Program of the Institute for the Science of Origins, a partnership of Case Western Reserve University and the Cleveland Museum of Natural History. It has been brought to you with the generous support of Richard Morrison and the assistance of Case Western Reserve University's College of Arts and Sciences, Media Vision, and WVIZ PBS and 90.3 WCPN IdeaStream. For more information on the Origins Science Scholars Program, including a full video archive, please visit the Institute's website at www.case.edu/origins.